We're so pleased to have you today. We have with us Mohammad Pourhosseinzadeh. A little bit about him. He is a seventh-year MD PhD student at UC Davis who completed his PhD in the lab of Dr. Mark Housing, where he studied the crosstalk between beta and delta cells. Today, his talk is titled Heterogeneity and the Coordination of Delta with Beta Cells is driven by both paracrine signals and low-density CX36 gap junctions. Thank you so much, Mohammad, for joining us today. I know you have a packed schedule, and it's really fantastic that you're joining us and walking us through this really interesting and elegant data. So thanks so much. And you can drop a question in the chat, and we'll take them at the end of the talk.
Thank you very much, Monica. My name is Mohammed. I was in Mark Housing's lab. I actually recently got my PhD in March, and I'm going to be talking about my dissertation project, which has been focused on trying to understand how beta and delta cells communicate with each other and coordinate their activity in response to elevated glucose. So the islets of Langerhans are primarily composed of three main cell types: the insulin-secreting beta cells and glucagon-secreting alpha cells, which act to lower and raise blood glucose respectively, as well as the delta cells, which secrete the hormone somatostatin that acts locally within the islet to attenuate the activity of beta and alpha cells. And so my project has really been focused on looking at the beta cells and understanding how beta cells signal to the delta cells to potentiate this negative feedback loop whereby beta cells, by activating delta cells, potentiate the secretion of somatostatin that acts on the beta cells to attenuate their own secretion of insulin. And so we know that this mechanism is important in normal physiology. This was actually a paper that our lab published in 2024 in Nature Metabolism, which demonstrated that if you get rid of the delta cells or you get rid of the somatostatin being secreted by delta cells, you can see the shift in the glycemic set point of these mice, showing that delta cells are actually important in establishing the glycemic set point within the islet. We also know that in diabetes, where you have a loss of functional beta cell mass, the loss of the signaling that normally occurs between beta cells and delta cells leads to delta cell dysfunction. And this dysfunction, in turn, leads to further dysfunction within the islet. And actually, there was a really great paper from Linford Bryant's group, it was Thomas Hill, just kind of showing how the loss of this communication can lead to aberrant sort of glucagon secretion when it's inappropriate. And that actually makes it harder for diabetic patients to regulate their blood glucose. And so we've known for quite some time, though, that beta cell insulin secretion and delta cell somatostatin secretion are coordinated over time. And this is just a graph from Salehi and Hellman's groups showing this secretion over time of somatostatin and insulin. And we can see from this that both are oscillatory over time, and that if you look closely, actually, somatostatin is trailing insulin secretion by about 30 seconds. And so we saw this data, and knowing this data that's been in the literature, we thought, well, this delay is really consistent with the sort of timescales that we associate with paracrine signaling. And actually in 2018, Linford Bryant had put out a really great paper that demonstrated that if you just optogenetically activate a beta cell, you can see delta cells turning on within about 30 milliseconds. And they showed that this was mediated via gap junction coupling. But of course, this sort of creates a bit of a conundrum and sort of a paradox. And that is, well, if you have activity or electrical activity that's happening at the order of milliseconds, and yet secretion seems to be delayed on the order of seconds, what exactly is the nature of that signaling? And so that really brings us to the two models that we sort of use or attempt to use to describe beta and delta cell crosstalk. And that's firstly, so paracrine signaling, whereby beta cells secrete a diffusible paracrine factor that acts locally on neighboring delta cells to potentiate somatostatin secretion, and also gap junction coupling, whereby beta cells are able to directly stimulate neighboring delta cells via direct depolarization. And so going into this project, I hypothesized that the predominant communication is paracrine signaling. Of course, gap junctions are serving a role, but that it's potentially paracrine signaling that's really mediating that delay that we see, at least at the level of secretion. To sort of establish what exactly is driving delta cells as we sort of go into this, and also establishing the model that we use. So we image islets in order to sort of assay their activity. And so for us, we use calcium imaging using GCAMP6, and we use calcium as sort of a proxy for secretion, since we know that an increase in intracellular calcium is necessary for secretion. And so this is just a video of islets from a mouse that's expressing GCAMP6 selectively in delta cells. The red nuclei that you're seeing in the image are actually the nuclei of beta cells. In this trace, you're going to both see and hear changes in calcium and in sort of intracellular calcium concentration. And what you'll notice is as we're in low glucose, the delta cells are quite active. And what we wanted to do here was establish whether or not this activity is dependent on depolarization. So we add diazoxide, which is a K2B channel opener, and isradipine, an L-type calcium channel blocker. So I'll let the video play. And so this is an intensity plot, which is just another way to visualize the data that I just showed. Every row in this plot is an individual cell, and every column is a point in time. Superimposed on top of the plot are line graphs, just of some representative cells. And what I hope to sort of demonstrate here is again, delta cells are quite active at 5.5 millimolar glucose, which is sort of the baseline that we use for beta cell imaging. It's just below beta cell threshold. And these delta cells, when they go into diazoxide, which you can see here in panel B, where I quantified the area under the curve, there's a significant reduction in their activity. And likewise, in isradipine, there's also significant reduction in their activity, suggesting that these cells, just being active in low glucose in the absence of beta cell activity, depend on some level on depolarization. And so going one step further, we also wanted to do the same experiment, but this time also looking at beta cells and doing this in high glucose, where we know beta cells and delta cells are coordinated and active. And so this is another intensity plot. In this intensity plot, the beta cells are at the top in green and the delta cells are at the bottom in red. And again, what you can see is that as we move from high glucose or 16.8 millimolar glucose to diazoxide, there's a significant and sharp reduction in both beta cell and delta cell calcium activity. And again, as we wash out the diazoxide and then add in isradipine, there's a sharp reduction in both beta cell and delta cell calcium activity. So what this again demonstrated to us is that most of this activity that we're seeing in delta cells, both in low glucose, so sort of beta cell independent activity, and also the activity that we see in high glucose that's sort of going on along with beta cells, is dependent on depolarization. And so we had this model now where we could image beta and delta cells simultaneously within the same islet. So these islets are expressing GCAMP6 in both beta cells and delta cells. And again, the beta cells in these islets are expressing a nuclear red fluorescent protein. So their nuclei are red. And I have two arrows here just to sort of orient us on two different delta cells. But again, in this trace, you'll be seeing and hearing the beta and delta cells as they're responding in real time to high glucose. So I'll let this play. And so this is just a still image of this islet, just to show all of the cells active. And because both the beta cells and delta cells are expressing the same fluorescent calcium reporter, we then do staining afterwards for insulin and somatostatin. And so this is just an overlay of that same staining to show how well we're able to sort of cross-reference those cells and confirm their cell type identity afterwards. And so this is an intensity plot of that same video. Again, beta cells are in green, delta cells are in red. And I think, and what initially struck us the most is that when we looked at this data, beta cells have this very clean, slow oscillation that's coordinated across all the beta cells. And that's expected because beta cells, we know, are tightly gap junction coupled to one another, and that leads to this tight coordination. Delta cells, however, have a bit of a mixed picture. We see both this coordination of these slow oscillations that are sort of appear to be coincident with neighboring beta cells. And at the same time, delta cells are actually quite active when beta cells are inactive. So during the nadirs in beta cell oscillations, you can see quite a lot of this uncoordinated, faster spiking. And also in 2.8 millimolar glucose, which is actually even lower than what I showed before, delta cells are again quite active, where beta cells are completely inactive. And again, this was important to us because earlier in sort of looking at these cells in response to diazoxide and isradipine, we had sort of come to the conclusion that most of this activity is largely depolarization dependent. And so it was interesting to us that we were seeing calcium activity when beta cells are inactive, knowing that beta cells and delta cells do share these gap junctions, much like beta cells are gap junction coupled to one another. And actually, when we focused in on the delta and beta cells on these slow oscillations, we saw that, interestingly, delta cells appeared to be turning on slightly ahead of the beta cells. And so to really dig deep into and quantify this coordination that we were seeing, I used an algorithm, or I created an algorithm in Python based off of work that was done by Slack-Rupnik's group. And essentially what I was able to do was sort of deconvolve or pull apart the slow component of these traces in delta cells from the faster uncoordinated calcium spikes. And so what this data here, or what this graph here is representing, at the top is the raw data of one of the cells that I showed earlier, and then effectively just a low pass filtered version. So any activity that's happening on the order on a time scale greater than 20 minutes, I refer to as my baseline. Below that is effectively just a band pass filtered version of my data. So these are features that are occurring on a time scale between 1 minute and 20 minutes, which I call my slow component. And then below that, I call my fast component. This is just a high pass filtered version of my data. So any events that are happening on a time scale faster than 1 minute. And so overlaying these graphs, we can see you have these slow oscillations, which look a lot like the slow oscillations that we saw in beta cells. And again, these faster uncoordinated calcium transients, which are largely occurring during these nadirs in the slow oscillations. And so the same filtering was applied to the beta cell data. And again, you can see here that beta cells have these slow rhythmic oscillations that correspond to the slow component, which are very similar to the delta cells. But beta cells also have a fast component. Importantly, the fast component that I pulled out from the beta cells are actually fully coincident with the slow oscillations. So these are faster spiking potentials that are occurring on top of the slow rolling oscillations in beta cells, which is different from what we saw in the delta cells. And so taking this data, what I was able to do was, again, pull out the slow component from the fast component and analyze the slow component of the beta cell and delta cell responses in isolation. And so what I was able to do here was measure the rate at which these cells are turning on and turning off, and also measure at what time point a beta cell is turning on and at what time point a delta cell is turning on. And so this is all in high glucose here. What I noticed is that the on rate for delta cells is faster than it is for beta cells, and the off rate is also faster for delta cells than it is for beta cells, just looking at the slow component. And then when I looked at the time delay, so this is looking at how much time passes on average between the beginning of one of these slow oscillations in a beta cell and the beginning of one of these slow oscillations in a delta cell. So the green graph here is actually just looking at their pairwise comparisons of beta cells. What this is showing is that on average, an individual beta cell will be turning on about or around the same time as a neighboring beta cell. We did the same thing looking at delta cells. These are pairwise comparisons of delta cells. And we saw that again, if you compare the time points at which a delta cell is turning on and the time point at which another delta cell is turning on, there's very little delay between them. Importantly, there's a larger spread in the data, and this is sort of reflective of the fact that delta cells themselves are much more heterogeneous in their response. But when we looked at beta cells and compared them to delta cells, we saw that consistently the average response of the delta cell was about 17 seconds faster than the response of a beta cell, which was consistent with what we saw visually just looking at the data 2 slides ago. And interestingly, when we looked at KCl, so this is just direct depolarization with 30 millimolar KCl, we saw a similar pattern. In this case, the on rate and the off rate of beta and delta cells were not so different, but the time delay was very much consistent. So we saw, again, a time delay of about 17 to 20 seconds with delta cells preceding beta cells. This, of course, started to bring up some questions, which was, well, if delta cells are turning on before beta cells, then who's really driving this coordinated slow oscillation? And so to get at that question, we actually used a new mouse model. So in this mouse model, we're expressing both the GCamp6 and TD tomato selectively in beta and delta cells. And what we're able to do here is, using the constant red fluorescence of the TD tomato, we can fax purify the beta cells and delta cells in isolation and image their responses in high glucose in the absence of other cell types. And so this first graph here is an islet expressing GCAMP6, or this SALSA6F construct in beta cells. And what we see is that beta cells, they give these very characteristic slow oscillations that are coordinated with one another. When I do a power spectral analysis of these beta cells, we see that there's a peak right around 5 to 10 minutes, which is reflective of frequency of oscillation of these slow oscillations, which is what we would expect. And when we FACS purified these beta cells, we saw again slow oscillations in the beta cells. Importantly, the beta cells are no longer coordinated with one another, and the oscillations themselves have different frequencies, and they sort of have different durations. But when we looked at the power spectral analysis, there's still a peak right around 10 minutes, which again is reflective of the fact that these cells, even in the absence of other cell types, just on their own, maintain these slow oscillations. Now, delta cells, delta cells in an intact islet, they have, again, these slow oscillations. When we did a power spectral analysis, we see a very nice clean peak right around 5 to 10 minutes, which is again reflective of these slow coordinated oscillations between beta cells and delta cells within a single islet. But when we FACS purified these delta cells, we noticed that these slow oscillations were completely gone. These delta cells were either inactive in high glucose, or the only activity that was left over was these faster uncoordinated spiking calcium transients, which looked very similar to the spiking uncoordinated calcium transients that we saw during nadirs in delta cell calcium oscillations. And so again, a power spectral analysis of this data demonstrates that it's essentially a flat line. That peak that we saw around 5 to 10 minutes, or a period of 5 to 10 minutes, is completely gone. And so what this suggests is that these slow oscillations that we're seeing in delta cells in an intact islet is really being driven by beta cells, despite the fact that these cells appear to be turning on earlier than neighboring beta cells. So bringing us back to sort of the central question of the paper, which is what is the role of paracrine signaling and gap junction coupling in coordinating beta and delta cells? We first decided to look at paracrine signaling, and in order to assay paracrine signaling without affecting electrical activity within the islet, we used a compound called MO141, which is a Rho GTPase inhibitor. What it does is effectively stabilizes the cortical actin cytoskeletal network, and that essentially prevents vesicles deeper down from the reserve pool from making their way to the surface, giving off their content. So this effectively inhibits secretion within the islet as a whole. And so this trace will go from low glucose to high glucose. Again, you'll see and hear these coordinated calcium oscillations in beta and delta cells. And as we move into MO141, you'll notice this coordination is lost between the two. But importantly, beta cells remain highly coordinated. I then wash out the MO141 in high glucose, and then wash out in low glucose with ghrelin and KCl at the end. And so this is an intensity plot of that same data. And again, what I really wanted to show here is that beta cells and delta cells, as we move into high glucose, you can see they're highly coordinated with one another. Most of the delta cells, at least, are highly coordinated with the beta cells. And as we move into MO141, there's a bit of a delay, but ultimately many of the delta cells demonstrate a change in their behavior. Either they lose their activity or they lose their coordination with neighboring beta cells. And importantly, beta cells themselves are unaffected by MO141. Their calcium oscillations are just as coordinated with one another, and there's no difference in their activity. As we wash out the MO141 and come back into high glucose, you can see much of this coordination starts to come back in the delta cells, demonstrating that this effect is reversible. And so this is just a quantification of that. So at the top here is looking at the cross-correlation value of individual delta cells relative to the islet average response. And we can see that delta cells have an average sort of cross-correlation value that sits at around 0.75, which means most of the delta cells are pretty highly coordinated with beta cells. But of course, there's a large distribution because not all delta cells are coordinated with beta cells. And as we move into MO141, there's a dramatic shift and drop in this average cost correlation value, again reflecting these delta cells losing some of their coordination with neighboring beta cells. And as we wash out the ML141, you can see the cross-correlation values return back to their pre-ML141 state. And so looking at beta cells, again, beta cells are completely unaffected by ML141. They sit right around a cross-correlation value of 1, which indicates that they have almost perfect correlation with one another. And that doesn't change when we move into ML141. It doesn't change as we wash out the ML141. So addressing the second half of this question now, looking at the gap junction coupling between beta and delta cells. So we know from our own data that connexin 36 is the most highly expressed connexin isoform within the islet. And in fact, it's the most highly expressed connexin isoform within delta cells. And so here in panel A, this is just a graphic from our data set. So these are bulk RNA sequencing data of alpha, beta, and delta cells. And we can see that beta cells have the sort of largest expression of the largest RNA transcript for connexin 36 in the cells, and delta cells have about a 10-fold lower level of RNA transcript. And so kind of going one step further and looking at the actual protein expression and localization within these cells, I was able to stain for connexin 36 in islets. So these are cryosectioned, sliced pancreas from mice that are expressing a membrane localized TD tomato. So the red fluorescence that you're seeing here is just outlining the membrane of these cells. Beta cells are stained in blue, delta cells are in white, and the small green puncta that you're seeing co-localized with the red membrane are connexin 36 puncta. And so what I was able to do was then quantify this. So looking at these puncta and then measuring the number of these puncta that are on the surface of beta cells and on the surface of delta cells, what I was able to quantify is that beta cells on average have about 9 connexin 36 puncta per 100 microns squared, which was in good agreement with the cryo-EM data collected by Palomeda. And also for delta cells, we saw there are connexin 36 puncta on the surface of these delta cells, but importantly, there's a significant drop in the average number. So for delta cells on average, there's about 2 of these connexin 36 puncta per 100 microns squared. And so this initially we thought likely reflects a decrease or a difference in the sort of abundance of connexin 36 on the surface of these cells. In panel D, what I did was actually go back and I looked at, so for every beta cell, I asked the question, well, what proportion of these connexin 36 gap junction puncta on the surface of a beta cell is on the plasma membrane surface that's neighboring an adjacent beta cell. And so if we're looking at a beta cell, almost 99% of the time, a gap junction on the surface of the beta cell is neighboring another beta cell. But when I looked at delta cells, about 30% of the time, one of those gap junctions on a delta cell was neighboring an adjacent beta cell. And about 60% of the time, it was actually neighboring another delta cell. So here we did sort of the gold standard experiment, which was getting rid of connexin 36. So for this experiment, what I did, I was kindly given mice that have a flox connexin 36 allele. This was given to us from Dr. Awatramani in Canada. And so these islets these mice, we were able to breed with our SST-CRE mouse line in order to selectively knock out connexin 36 in delta cells. And what we noticed here is that when we knocked out connexin 36 in delta cells, we largely saw a reduction in the coordination between beta cells and delta cells, and largely a reduction in the slow component of the oscillations in beta and delta cells. And so this is just a quantification here of this data looking at these SST-CRE flox connexin 36 mice and wild type control mice. And what we see is that for beta cells, there's a slight reduction in the cross-correlation value. However, it's very, very mild. And for most of the beta cells, they're still very highly coordinated with one another. And for delta cells, there's a very significant and dramatic drop in their cross-correlation value, again, suggesting that gap junction coupling is necessary for the coordination between beta cells and delta cells, that it is playing a role. And that specifically it's connexin 36, the connexin 36 isoform that's forming these gap junctions. And so coming sort of to the end of this, what I noticed in sort of imaging all of these beta and delta cells, or all of these delta cells, is that delta cells, unlike beta cells, are not this sort of monolith. Delta cells, one delta cell is very different from another delta cell, and actually there's a very rich tapestry of activity that I saw just in imaging delta cells. And so what I did here was I tried to sort of categorize those differences. And so these are delta cells from about 4 mice I think about 8 islets. So this is across many mice, many islets. And what I saw consistently is that I could sort of separate delta cell response into sort of into a couple categories. So first, based on whether or not they were active in low glucose, and then secondly, whether or not, or essentially how coordinated they are in high glucose. So I sort of separated this into 3 categories: delta cells, which are synchronous with beta cells. So these are delta cells that do not have these faster uncoordinated calcium transients during nadirs and beta cell activity. And oftentimes these delta cells also don't have a whole lot of activity in low glucose either. They're very beta cell-like in their response, but they are delta cells. There's also delta cells that I saw, which I categorize as pseudosynchronous. These are delta cells which, much like the beginning, what I was describing, they exhibit these slow calcium oscillations, but superimposed on top of those slow oscillations are also these faster uncoordinated calcium transients. And then lastly, delta cells, which I categorize as uncoordinated. These were delta cells that, despite being in an intact islet, at no point during high glucose demonstrated any of these slow coordinated oscillations with neighboring beta cells. And actually there was a 4th category here, which were delta cells which were inactive in high glucose. And I want to point out, these are delta cells. This is a very, very small population. These are not sick delta cells. These are living delta cells. They responded normally to ghrelin, which is a delta cell specific activator, and they depolarize perfectly fine to KCl, but they're inactive in both low and high glucose. And so taking one step back at this point, I also wanted to ask the question of, well, is this heterogeneity that we're seeing in response to delta cells in some way a reflection of how these delta cells are coupled to neighboring beta cells within the islet? And so looking back at the data, my connexin 36 gap junction staining, what I did is I quantified the proportion of cells that have 1 or more gap junctions on their surface compared to the proportion of cells where I didn't see a detectable gap junction. And so what I want to point out here is that even for beta cells, there's quite a large, actually the majority of the cells that I imaged in these slices, they didn't have any observable puncta on their surface, but this was staining that I did in a sliced configuration. And so, looking at just a sort of 2-dimensional slice in what is a 3-dimensional structure, we're sort of undersampling what parts or how many of these cells actually have gap junctions on their surface. But despite that, comparing just the proportion of beta cells with gap junctions on their surface compared to the proportion of delta cells with detectable gap junctions on their surface, there's a significant difference between the two of them. Beta cells, about 40 to 50% of the beta cells I imaged in these sliced preparations had at least one detectable gap junction on their surface, and about 10 to 15% of delta cells had a detectable gap junction on their surface. And so we think that this difference that we're seeing between beta cells and delta cells is most likely reflecting a difference in sort of the populations of delta cells, suggesting that there may be some delta cells that don't actually have gap junctions. And that would be consistent with the behavior that we see, that there are about a quarter of these delta cells, almost 25% of them, are actually completely uncoordinated with neighboring beta cells. And they may represent a population of delta cells that just don't have gap junctions or are just not coupled to beta cells in the same way as the other delta cells. And so that brings me to the last slide, which is just sort of a schematic of how we think beta and delta cells are communicating with each other. And that is that delta cells are this sort of heterogeneous population of cells, and that some delta cells are very reliant on gap junctions, connexin 36 gap junctions for their coordination, that many delta cells depend on varying levels on paracrine signaling via a still unknown paracrine factor for their coordination, and that some delta cells may not use either gap junction coupling or paracrine signaling, and that they may actually be coupled or communicating in a different way, or maybe not communicating with the beta cells at all. And so I'd like to thank everyone in my lab, the undergrads who were actually really important for this project and sort of collecting all the data and making this happen, as well as the former lab members in the Santana lab and Contreras lab at UC Davis who have been really helpful in sort of understanding gap junctions and doing a lot of this work. And so I want to thank everyone in my sources of funding, and I'll take any questions now. Thank you very much.
Thank you, Mohammad. Beautiful talks, more beautiful work coming from the housing lab at UC Davis. I have a question from Matt Marins. What are the mechanisms underlying the delta cell electrical activity at 5.5 G? I.e., how does the membrane voltage get high enough to reach the threshold for voltage-dependent calcium channels? He can unmute if he'd like to.
Yeah,
Matt?
that's a really good question, Matt. So I think there's a couple ways that I sort of look at delta cell electrical activity, I guess, and sort of how it's different or fundamentally different from beta cells. And one of those is that delta cells have a much higher membrane resistance. More of those K2P channels are closed most of the time relative to beta cells. And so
more like alpha or similar to alpha. I mean, where does it sit? I mean, in alpha, I think of, the number that Roersman gives me, which is 99% of the K2P channels in alpha cells are closed. Is it similar in delta?
I don't remember the exact number. I have to go look actually at Roersman's papers too, because I think that's where I'm getting my my idea from. Yeah, I don't remember the exact numbers, but I do know it's significantly higher, and I think that's actually part of the reason why we see a lot of this uncoordinated activity even in high glucose when beta cells are inactive, and also partly probably why delta cells are turning on slightly earlier than beta cells. I think delta cells in general are just more excitable. I think they need less to sort of tip them over. What exactly is tipping them over? I'm not entirely sure if it's the small amount of glucose that's in the media. I will say that I've done experiments with delta cells just doing a glucose step, so starting from 0 millimolar glucose and making my way up. And delta cells, at least in the experiments that I've done, they're inactive in 0 millimolar glucose. So I think there is some level of sort of glucose metabolism in there. Also, delta cells don't have GLUT2 on their surface. I think it's GLUT1. So there's a difference in sort of KD, right? Like those GLUT2 receptors have a much higher threshold for bringing glucose into the cells, and that might be part of what's leading to this difference. So yeah, I guess like a long-winded answer to what your question is. I think the delta cells are probably responding to lower concentrations of glucose because they're able to take in that glucose, and also they're just generally more excitable, and so it takes less to get them going. That's sort of my interpretation.
Yeah, the high membrane resistance argument makes sense to me. The surprise that I got, at least, I have not patched a delta cell, but in patching alpha cells, what we found is that the membrane potential is still pretty low. I mean, certainly they've got the higher membrane resistance, which I agree with Patrick's idea that, that makes them easier to get to threshold. But I was surprised at how low the absolute voltage actually was. I mean, we're still talking about like minus 68 or something like that, or minus 65, which is nowhere near the threshold for voltage-dependent calcium channels. And so I'm just confused as to why we're seeing that. And it sounds like the delta cell has a lot of that same characteristic with the alpha cells. It seems oddly like, I mean, the beta cell almost seems like the outlier at this point.
Yeah.
Of the three endocrine cell types.
Yeah.
I have more questions, but I don't want to take away from others. So you can circle back to me if there's time.
Sure.
Yeah, well, we love questions. I have a question coming from David Hill. Does the coordination of oscillations between beta and delta change with aging in mice?
That's a great question. I've done a couple experiments with older mice. I think the oldest I've ever gone is like maybe 1.5 years or a year, and I haven't seen a difference. I haven't done a sort of exhaustive review of that quantitatively, but just visually, based off the experiments that I've done, they still seem to be just as coordinated. They look very similar to the younger mice between 3 to 6 months old, which is what I used.
Okay, cool. I had a question. Pedro Herrera just had this paper coming in Nature Metabolism, August 21st of 2024. And, the title was Regulated and Adaptive in Vivo Insulin Secretion from Islets Only Containing Beta Cells, sort of implying that, all you need is beta cells. And what would your sort of comments be around that? I mean, we talked briefly about this just offline, but I think those comments that you shared were really illustrative.
Yeah, so Mark just popped into the chat and said part of it, but so in his paper he ablated both alpha and delta cells, and I believe showed that there was no change in the sort of glycemic set point, and this is in response to just challenge with high glucose, which we were talking about, sort of how these cells may also play an important role in sort of mixed meal stimulation. I mean, what happens when you have proteins and fats in the equation, but also, and Mark pointed out, in the supplementals, he did a very similar experiment to what we did in our 2024 Nature Metabolism paper, which was ablating the delta cells. And he saw that when you ablate just the delta cells, you do see a reduction, a decrease in the sort of glycemic set point of these mice. And so I think the question that comes to mind, and sort of the way that I rationalize this is, alpha cells in the islet are in the context of high glucose, largely acting to potentiate and activate beta cell insulin secretion. Delta cells are largely acting to suppress that secretion. And so it might be the situation where when you get rid of the gas pedal and you also get rid of the brake, that you don't really see a change, but that doesn't necessarily mean that the gas and the brake aren't important. I think they're still important and they're still playing a physiological role. But so that's my interpretation. Yeah, great question. Thank you.
Yeah, thank you. And I guess I would also just add, like, obviously delta cells have been shown to receive the cholinergic input from the parasympathetic nerves. Some are saying, it's the vagus that brings that. And I wondered, like, okay, what's your hypothesis or thoughts around how that also adds to this symphony?
So the parasympathetic signaling within the islet. So I guess just from a physiological standpoint, we know that if you have parasympathetic activation, you'd be sort of in a rest and digest state. And so you would want beta cell insulin secretion. Actually, so yeah, and I'm thinking of this actually more from the sympathetic angle where you have, I actually, I want to say, I want to think about that more actually a little bit.
It's just kind of like another level of complexity, right? Like they respond to acetylcholine and other neurotransmitters. So then just like, okay, it's like one more level that you have to sort of think about, one more set of experiments that, you probably could bring your beautiful system to. Matt, did you want to have another question?
Yeah, sure. Well, actually, just one question on that before we leave acetylcholine. Will that light up a delta cell? I know it makes the alpha and beta cells go berserk from a calcium perspective, but does it also light up the deltas?
Yeah, so acetylcholine, I've used carbachol, muscarinic agonist. So
yeah, sure.
That one, just thinking about the experiments that I've done. So that one, when I add carbachol, is a very interesting response because it has this initial activation, so sort of GQ mediated, and then suppression that's sustained, which is like a GI. So and I don't know if that's just a dosage thing. Maybe the dosage of carbachol that I used was too high, maybe not biological.
Presumably that was in the intact islet as well, right?
Yes, it's in the intact islet. Well, so there also could be paracrine.
Right, so some of it can be paracrine also.
Yeah.
It's really hard to know what's going on unless you've got ML141 present during that experiment.
That's true. Yeah, I haven't done at ML141. I guess I was thinking more from the perspective of having M2, M4, and then also M1, M3 receptors on the surface of the cell acting in concert with one another. But yeah, it totally could be paracrine signaling too.
Okay, well, that wasn't what I wanted to ask, but I'm wondering if you can clarify for me something about the structure of the gap junctions. When you knock out a gap junction in a specific cell type that goes to another cell type, for example, the beta to delta connections, can you knock that out from either cell type? Like if you lose one on one side, you also lose the connection between the two?
Yeah, my understanding.
If you knock it out in deltas, do you lose beta deltas? And do you lose... I could see you losing delta deltas, but do you also lose beta deltas?
Yeah. My understanding is that you would, right? Because the gap junctions, you have to have the expression on the surface of both cells to form a functional gap junction. I guess the other, if you lost it on one cell, then maybe you'd have a hemichannel on the other cell. But I think more likely whatever is left on that cell is probably going to be used or relegated to form gap junctions between other beta cells or other cell types that do have gap junctions. So my impression is that you would lose the beta delta connection if you got rid of gap junctions in just delta cells, because you wouldn't have a second half of connexons to form a functional channel. I don't know.
Well, what happens? Yeah, I know. It brings up this question of what happens if you have half of one.
If you have half of a channel?
Well, if you have you called it a hemichannel. If you have a half of it in a delta, but then there's no partner in the beta, is it totally dead?
Oh, you mean is it functional?
Yeah. In any capacity. I don't understand the structure of connexin 36, and I guess it's a structural biology and function question.
So I know. So there's been some debate. I've kind of digging into the literature. I don't think it's fully appreciated or fully thought that hemichannels even exist, period, on the surface of cells, or that they functionally have an effect. There are some labs. I think it was Palometa who showed or suggested that you might have hemichannels that can allow like ATP to sort of leave the cell. So a hemichannel on the surface of a cell would basically just be another way for the cell to let go of fat, like paracrine factors or something, out of the cell in a way that's exocytosis independent or independent of sort of vesicle trafficking. The sort of data there I think is a bit tenuous. I don't know that like physiologically it really serves a function. It seems like people are kind of back and forth there. But yeah, if you knocked it out, so in my experiment where I knocked it out in delta cells, I would expect you wouldn't have a functional gap junction or sort of connection between the beta and delta cells. You might just have a hemichannel on the surface of the beta cell, which would, in theory, give a pathway for the beta cell to sort of give off some paracrine factors or some factors. But I think more likely that gap junction, that hemichannel probably wouldn't really exist, and it'd just be a gap junction with another cell, like another beta cell.
It makes me wonder about, the piston-Benninger connexin 36 experiments. So there, if you delete connexin 36 in the beta cells, and that leaves, you've still got, the delta cell still contributed its half, but the beta cells is gone.
Yeah.
Does it go and find other delta cells and change the delta-delta coupling? Do you know what I mean? Is there an artificial rewiring of that we hadn't considered, or that they hadn't considered when they looked at that model that's important? Or can we accept all of the conclusions from the beta cell connexin 36 knockouts sort of as they were presented?
Yeah, no, that's a really good point. I think it's totally reasonable to think that you might have some level of rewiring. I guess I don't know what exactly is controlling how the wiring between delta cells is occurring, and if in not having those sort of beta cell partners, would you have more connections with the delta cells, or would you just have the same number but no longer have those connections with beta cells? So that's a really good point. Yeah, I don't know.
Could you guys do some imaging, like some, like super resolution imaging or some kind of really high level resolution imaging to look at tissues from those who have diabetes or post-diabetes onset, to look at the quality and quantify the types of gap junctions that remain on the delta cells? That'd be fairly cool.
Yeah, I mean, I think I could even just use the staining that I have. I think that would be a really good tool to sort of look at this question. If we had islets that had the gap junctions, which we actually do, islets with the gap junctions knocked out of beta cells, do we see more connections between delta cells or more of those punked on the surface of the delta cells? That would be, a good experiment.
Like a compensatory mechanism.
Yeah.
Well, there's a lot of work left for you, Mohammed, and very little daylight hours. So thank you again for, here's one last question. Sorry, Kiadi Gerder. Is there a physical connection or some protein factor secreted by one cell signaling the other cells? If you co-culture, would you see similar oscillations? I'm not sure if she's asking about paracrine impact.
Yeah, so I think we do think that there's some protein factor or something that's being actively secreted by the beta cell that's sort of helping to mediate this coordination. Oh yeah, if we co-cultured them, would we see a similar oscillation? Yeah. So I've thought about that. I've kind of given it a try. It's a bit challenging because as cells are releasing these factors, the sort of local concentration of the factor very quickly decreases over distance, and so it's a bit of a challenge to sort of have these cells cultured in a way where they're sort of far enough away that they're not forming gap junctions, but close enough that we could say whatever paracrine factor is being secreted is sort of acting on the downstream cells. It's a good suggestion and definitely something that we've thought of. It's just a challenging experiment technically to do, or at least it has been in our hands.
Yeah, there's so much. It's a very fertile area as you guys are dissecting out the mechanism of the physiology of these islet cells and how they all coordinate. All right, well, that was a great discussion. Thank you very much, everyone, for participating and joining. And thanks again, Mohammed, for making time to share this excellent work with us. Appreciate it, and wish you all the best. And we'll talk again soon.
Thank you. Thank you for having me.
So much. Bye-bye.